guys, welcome to Science and Tech series, and this is our wonderful Science and Tech series. Guys, we are going to coming. Yeah, we did space technology, right? Now we are going to doing this biotechnology. Okay, please be attentive and please remember everything what I am saying in the class, and it will help you to crack the exams. Don't try to cram everything, just try to listen careful and if want write down it in your notes and otherwise leave and but listen carefully and read it twice, thrice for how much time it possible that much time you have to read. Okay, don't take see neglect while exam is in the March, while the exam in the May. Don't take too much. It is very just we have 10 months, just 295 days only we have. Okay, so be careful and yeah, let's go to the learn biotechnology. Don't want to waste our wonderful time, right? Okay, let's go to what is the biotechnology. Biotechnology is use of biological organisms and cells. The biological systems are biological process that manufacture product and technology that improve the quality of human life. Anything we are using some biological things, biological any organism, cells and biological system or biological process, any related to the biological, we are adding some technology to manufacture something. For example, the curd. When we add, okay, for curd production, in that bacteria that is natural, right? When we want to make bread, that is we add yeast, right? And in the same way, we like that gene therapy, we divide and cut the specific point of the DNA for gene therapy, right? Like that, we are we use our technology to change anything. Okay, those in biological form, we add any technology that is the our biotechnology. Okay, I think you get like this. Here only there biotechnology, biology plus technology. Okay, that's that remember that only. For example, curd production, vinegar and test tube baby, gene therapy, bread making, alcohol, genetic modified crops, also cheese, penicillin, and DNA machines. Okay, classification of biotechnology. Here we have broadly two categories we classified. One first one is the traditional biotechnology, second one the modern biotechnology. Traditional biotechnology simply exploits the natural capability of microorganisms without changing their genetic makeup. Means what is happening here? Okay, classical traditional biotechnology, right? Traditional biotechnology simply exploit the natural capability of microorganisms without changing the genetic makeup. And in the same way, modern biotechnology involve manipulating the genetic material. We need to involve here manipulate the genetic material, that is our DNA, to obtain desirable traits. The traits means something character are wonderful. Okay, characters are produce valuable product. Okay, for example, characteristics here: no genetic modification. In old tradition, there is no genetic. We will not do any genetic alter. Okay, here, and uses naturally occurring microbes. Only we use microbes, bacteria like lactobacillus, right? Lactobacillus in curd making, right? And also old form of biotechnology. Oldest, it is the oldest form of biotechnology. It is based on the fermentation. Okay, fermentation occur in when like that our dosa flour, right? Right? We make flour for making dosa. There will fermentation happen. That bubble white because of the carbon dioxide release. And here, genetic engineering and DNA manipulation and cell culture or tissue culture. Cell culture means our own cells, they will culture it. It's, and tissue culture also same. They will take our tissue, they will do experiment out of take our tissue and experiment they will do in the lab. Like recombinant DNA technology also they will do. Okay, here only then we see. For example, tissue or cell culture, gene therapy, recombinant insulin, DNA vaccines, and GM crop BT cotton, curd, cheese, bread, alcohol, vinegar, penicillin. Okay, let's go comparing. Yeah, that only na comparing is it is natural microbial activity. It is altered genetic material. Here no DNA manipulation happen. Here DNA manipulation is involved. Here ancient technology. It is advanced technology. What we are using now. Okay, fermentation based. It is genetic engineered based. Curd, bread, beer, BT cotton, insulin, gene therapy. Basis of modern biotechnology. What are the basis of modern technology? The modern biotechnology became possible because scientists understood that almost every living organism contains DNA. Yeah, what are the living organism in the on the earth? They all are has DNA except our virus. Virus has RNA. Okay, but it is we consider it as non-living. Yes, important exception is there. Okay, carefully listen it. The most organism possesses DNA. Okay, most organisms have DNA. Exception, some viruses possess RNA instead of DNA. That only. So, for example, for that virus, tobacco mosaic virus and QB bacteriophage. Okay, because DNA can isolate it, cut, modified, and recombined, scientists can change characteristics of organisms. That is modern biotechnology. Okay, here DNA, our DNA they can isolate. Isolate means they will take out from the group and make it in separate, and they in lab they will modify something like they will change one gene from the DNA like that. Okay, you have to clear the definition of what are DNA and what is gene, what are gene and RNA what is. You have you have a clear clarity on about it. If any question asked in the exam, you can crack it. Here gene and gene mapping. Okay, what is gene here? A gene is a basic physical and functional unit of heredity, and it is a specific segment of DNA that carries instruction to produce protein. That carries instruction to protein. Okay, let me show the what is the gene here. Here there is no diagram. Okay, sorry guys, I didn't include here diagram. Okay guys, now gene and gene mapping. Okay, we studied, right, this modern biotechnology. What is that? There is no we have to modify it in some biological form of livings or anything organisms or anything into by adding our modern technology. We need to modify that organisms into that is useful for the our human being. Okay, and for example bt cotton, right, bt cotton we have modified, and also mustard, right, and brinjal also, right. Now okay, what is it is a gene, right. Here they are what they are saying about gene is the basic physical and functional unit of hereditary. It is a basic physical functional unit of hereditary. Yeah guys, it is very uh, it is very functional unit of our life because this is DNA. Let's assume, okay, this is DNA like that, right. Oh sorry, it is taking huge time. What has happened? Okay. Okay, this is DNA. Okay, this is gene. Okay, this is gene. Okay, just assume this one turn of our DNA, one unit of DNA, that is our gene. It will has our entire character, what are we are speaking, and our hand color, our space structure and shape of and height, all these will depend on these genes. Okay, if any defect in this part of gene, it will may lead to some disorders. That's why gene therapy has came. Okay, that's why biotechnology is very important. Basic and functional unit of hereditary. It's a segment of DNA that carries instruction to produce protein. Yeah, that will also help, right? Means when DNA undergo to the RNA undergo in the form of translation. Okay, and translation when RNA to need to protein, then transcription will happen. Okay, from M messenger for mRNA means messenger RNA to we form protein. Okay, from RNA to with the help of amino acid and with the help of translation, we will produce protein. Just remember this. I will say what is happening in next. Okay, what are the transcription translation? I will say in the next one. What are the important feature of gene? Okay, it is located on DNA. Just remember, gene is located on DNA. It is a functional unit of hereditary. Okay, and control inheritance. It will control inheritance. Means from mother character to offspring character will expose. That is inheritance, right? It will transferring the character from mother to child and determine physical traits. Okay, it will determine our physical traits, height, right? Height, color, like that, and code for proteins. Yeah, these that only recommend, right? For RNA, for DNA, that will recommend to code for protein. What is human genome? Yeah, like what is that? According to Human Genome Project, human possesses approximately 20,000 to 25,000 genes. Just remember, in Human Genome Project, human has approximately in one DNA 20 to 25,000 genes are there. Assume guys, cell, under cell, under the nucleus. In that nucleus, we have chromosomes, and in the chromosomes we have DNA, right? In that DNA, like here, 20 to 25,000 gene we are there. That is Human Genome Project, right? We have identified from the human genome project. Okay, it's non-coding region make up a huge portion of the total human DNA. That like okay, gene mapping, guys. Just be careful here. What is gene mapping? Sorry, I did mistake right here. Not did any mistake. Okay, let's go. Gene mapping. Definition. Gene mapping is the processes of determining. Okay. Gene mapping is processes of determining. Any process will need to be determined. What is the position of the gene on a chromosomes and specific biological function? Anything we have to determine. That is, we need to identify gene mapping means if any defect in the DNA. Okay. If any disease happen and we need to detect that part of gene, and that part will expose if just assume this part of gene is affected. Okay, because of this part, our our whole body will act differently. Okay, means any symptom will be like sickle cell anemia and anything that is defected gene diseases occurred, then our complete body will depend on that, right? Like that, we need to mapping this gene, right? We need to mapping this specific part of the gene. This is gene. Okay, this is part of gene, specific the part of that gene. So be aware. Okay, that is gene and chromosomes. Okay, position of gene on chromosome only. DNA there in chromosomes, okay, and its specific biological function. It has its own specific and importance of gene mapping. What are the importance? We need to get the identification of disease, what are the disease is there, and genetic research if we can happen, and crop improvement in plant also, guys. Gene mapping also same plant, okay. Don't think if gene mapping we seen only DNA in animal like human being, okay. Don't think like that. Means DNA has in plant also, human being also, any organism present in the earth. And gene therapy, okay. Here, so okay, let's, what are the importance? Disease identification, okay, we can identify the disease and we can get to know the genetic research also, and also crop management, and also gene therapy. Crop management, not crop improvement. Okay, let's go to come to, we need to understand broadly, right? DNA, deoxyribonucleic acid, what is this? Deoxyribonucleic acid. DNA is the hereditary material present in almost all living organisms. Yeah, right. DNA is hereditary material. How, right, guys? Yeah, it is hereditary material. Just here, it is hereditary material present in almost all living organisms. Just remember, guys. Okay, it is material because sometime asks it will alter something, and already PYQ has uh, given from this. Okay, you can observe around 2019 or 2022 they have given. Let's go and observe there also. It carries genetic information from one generation to another generation. Let's there, from mothers to offspring, right? From one generation to next generation. We often don't think, okay, if anyone are not understanding this simple English also, please do read newspaper daily, and try to, if you don't know the meaning of the simple word, and go to google and know the meaning of that word. Means knowing, the developing our conceptual understanding in UPSC is play a very huge important role, otherwise we burning out in the whole preparation. Location of DNA, okay, most DNA is inside the nucleus. Wow, right? Now we are going to the study of DNA, right? Here location. Just remember, okay, it's present in the nucleus and chromosome. I said right. In chromosome, it is there. Small amount in mitochondria. Just remember, guys, mitochondria also DNA is present, and in nucleus also, called mitochondrial DNA. Okay, mitochondrial DNA inherited maternally. Next, here structure of DNA. What is that? It is double standard. Okay, DNA has double standard. I will show here. Let me show you guys. This is single standard and this is another single standard. Both has DNA. It is DNA. Okay, double standard. It is handed double helix. Right-handed double helix and polymer of nucleotides. And these are nucleotides, guys. These lines. Okay, these lines are nucleotide. Human DNA length is 3.3 into 10 power 9 base pairs. See guys, human DNA length is 3.3 into 10 power 9 base pairs are there. Nucleotide components. Hey, what are the nucleotide components? Nucleotide, no, right? Those line between the two helix. I will show. These only nucleotide. Okay. Three component of nucleotide is there. Nitrogenous base. What are, and they had three component. Okay, one is nitrogenous base, deoxyribose sugar, and phosphate group. Need to remember this. Okay, Nucleotide components are nitrogen base, deoxyribose sugar, and phosphate group. Nitrogenous base are base pairing rules. Nitrogenous base are base pairing rules. Here, purines and pyrimidines. Nitrogenous base. Here, just go through here, guys. In the string component, first one nitrogen base. Okay, in that nitrogenous base, this nitrogenous base, right? Nitrogenous base, purines and pyrimidine is there. Adenine and guanine are purines. The pyrimidines, cytosine and thymine and uracils. Uracil present in RNA, okay, instead of thymine. And don't confuse it, okay. Thymine is present in the DNA, uracil is present in the RNA. Okay. Please clarify in your mind here only. Don't deviate from source to source. And Charkaka rule for DNA base pairing. The pair T. A pairs with T, right? A is 2 and G pairs with C. G means guanine pairs with cytosine and A adenine pairs with thymine. Crucial UPSC Prelims fact. Yeah. Okay. See that only na. DNA present in the thymine, uracil present in the RNA. That only. Storage of genetic information. DNA store information through the sequence of 4 bases. Okay. DNA will store information, guys. DNA will store information through the 4 base pairs in that A, T, G, C, right? You know. Any changes in this sequence changes the protein formed called mutation. Okay, any changes happen is this bases pair of nitrogenous base, then mutation will occur. Means changes will happen means mutation is happen, right? Okay, sequence protein. DNA fingerprinting, yeah. No, DNA fingerprinting, what is that?